So just a short introduction about myself. I'm Jonathan Concepcion, and I am a research associate at Louisiana State University, and I have a degree in biology from Central Luzon State University, and I previously worked at the Philippine Rice Research Institute. And currently and previously, I'm working with developing rice varieties for climate change resiliency with tolerance to salinity and drought stresses. Okay, so for today, let's talk about experimental design and setup, which is, I think, one of the most important parts in your research projects. Okay, so first, let's define what is an experiment, since basically we will be dealing with experimental design throughout this presentation. So basically, it's a method of data collection designed to test your hypothesis under controlled condition. So ibig sabihin, you will collect data para makita mo kung yung bang assumption mo, yung hypothesis mo ay tama or mali. And then it is under controlled condition para walang external factor na makakaapekto sa experiment mo or walang external factor na makakaapekto sa hypothesis mo. Okay? And then there were two types of study. One is your observational study and the other one is your experimental study. So what's the difference between these two? So I just wanted to highlight this. So your observational study is that you will just collect and analyze data without changing the existing conditions. So you will just observe, you will just take down notes of what is happening, you will just see. So it's sort of nanonood ka lang. And then take down ka ng notes kung ano yung nakita mo, then that's it. While compared to your experimental study, you are deliberately imposing treatments or you are changing conditions on subjects and observing their responses. So you are manipulating the conditions para makita mo yung expected mo na output or para makita mo if your expected hypothesis will actually be the result. Okay? So why is it important to have an experimental design? So bakit kailangan mo pang i-design yung experiment? Hindi ba pwede na basta mo na lang i-change yung condition, basta mo na lang i-test yung uh, subjects mo? So first, it is to infer about cause and relationship rather than simply describing. Okay, so like I said, we're dealing with experimental studies and you're not just observing or describing a certain thing or a certain phenomenon. Okay, and you wanted to know what causes this type of response, what causes this type of relationship uh, in relation to your objective and to your hypothesis. And of course, you wanted to gain information without ambiguity. So, ibig sabihin, gusto mo makakuha ng data, gusto mo makakuha ng information na malinaw. Okay, that's why it needs to have an elaborate design for possibility of alternative relationships, consequences, or causes. And so, ibig sabihin, gusto mo ring tingnan ano pa yung ibang relationship na andon, ano pa yung ibang factor na magko-contribute doon sa uh, experiment mo or dun sa uh, hypothesis mo, something like that. And of course, rule out alternative causes, leaving only one factor, that is the real cause. So, again, i-rule out mo yung ibang dahilan or yung ibang factor para yung gusto mong i-test, yun lang yung maitetest mo and yun lang yung makaka-affect doon sa study mo. Okay? Leading to your results and leading to answering your hypothesis. Okay? So this is just the basic flow of research that we follow. So first is we have to define the problem. So if you're here in the first lecture of Sir Jonas, he actually described this one really well. So first you have to define the problem. Ano ba yung gusto mong mangyari? Ano ba yung gusto mong pag-aralan? Something like that. Then determine the objectives. From that problem, from that study, ano exactly yung gusto mong makita? Determine the responses and the factors. So what are the data that you need? What are the conditions that you need to impose? Uh, so that's the important thing that that you need actually, and it has to be related to your objectives. Then you have to design your experiment. So your experiment should be your way to answer your objectives. Okay, and then your experimental design will actually dictate how are you going to measure these responses and how are you going to control the factors. Oh, sorry. Okay, next is of course you need to conduct your experiment and collect your data. So again, this lecture is only for designing your experiment. We have a different lecture probably in 2 weeks time for collecting data. Then, of course, you need to analyze it to make sense of the data that you got. Kasi, of course, puro numbers lang yung makukuha mo when you collect your data. Now, to make sense of it, you have to analyze it. So once you analyze it, you have to interpret. Ano ba yung implications ng data na nakuha mo? Ano ba yung implications ng result ng analysis mo? Then you have to verify your predicted results. This is where you now test your hypothesis. So did your data actually back up your hypothesis? Did your data contradicted your hypothesis? And then based on your results, what are the future experiments that you need to do? What are the future plans for your research? Or do you need to repeat the research? So this is the entire flow of your research project or when you conduct the research. Okay, so now we're going to the important terms that you need to remember when you design an experiment. So first is the subject. So it's the experimental unit or entries to which your treatments are imposed. So this could be your human subject, this could be your plants, this could be your bacteria, this could be your mice. Okay? So basically, ito yung subject ng pinag-aaralan mo dun sa study. Okay, this is to where your conditions will be imposed. Next is your factor, which is your controlled independent variable, and it is the category of treatments. Okay, so since we're here already, let's talk about treatments. So it's the condition exposed to your experimental units, and it is usually done by levels. So later I will differentiate the factor in the treatment. And then, of course, the response are the outputs or measurable outcomes that you collect. So this will be your data, the responses. So just for example, take for example this study. So this was done by my previous students from Nueva Ecija High School. So they studied the germination and early morphogenesis in rice as influenced by increasing levels of salinity and drought stresses. So of course, our test subject is rice. So we have 5 different rice varieties for this study. Then these are the factors. Okay, so this is your controlled independent variable. Now, of course, the increasing levels will be your levels. So if you combine these factors and levels, this will be your treatment. So we have treatments of increasing levels of salinity. We have treatments of increasing levels of drought. And of course, the response is the germination and early morphogenesis of rice. So the germination and the early morphogenesis are the ones that we measured. Okay, so I hope that's a little bit clearer now. Okay, next is your variable. So these are the characteristics that can take on different values. So these are the different things that could play a big part in your experiment. Okay, first is of course your independent variable. So like I said, it's the cost, it's the treatment that you applied to your experimental group. Ikaw ang magseset nito. Okay? So gaano katindi yung treatment na ilalagay mo? Is it 5%? Is it 10%? Is it 15%? What type of treatment are you going to apply to your experimental units? So that's your independent variable. Of course, your dependent variable will now be the effect. So it's the response or the outcome of your experimental group which you need to measure. So nung nilagay mo yung treatment, ano yung nangyari? Humaba ba? Nagkaroon ba ng zone of inhibition if that's antimicrobial activity? Ano yung naging performance ng mga mice mo? So that's the dependent variable. Now, the controlled variable is the condition, okay? The quantities or items that should be constant and must be observed carefully, okay? So this is also another thing that you set. The conditions of your experiment. Saan mo siya ilalagay? Lahat ba sila nasa dark condition? Lahat ba sila under sunlight? Uh, lahat ba ng bacteria mo, for example, ay nasa 28 degrees Celsius? Or what temperature are you going to set it? So that's your controlled variable. So again, let's use this study as an example. So for the independent variable, of course, we have the concentrations that we set for drought, which is based on percent polyethylene glycol, and then the concentration levels of salinity stress, which is based on your electric conductivity levels of your sodium chloride solution. Okay. Now the dependent variable will be your phenotypic responses or the response of your rise, which is of course based on percent germination, the shoot length, and the root length. Now our controlled variable here are the conditions that we set. So we set so we put the Petri plates, which is of course of the same size so that there will not be any bias, uh, in dark condition at 28 degrees Celsius. So this should be maintained, which is why I said in the previous slide that you have to really look, that you have to monitor it or something like that. And of course, we also control the varieties of rice that we use and the amount of treatment or the amount of solutions that we give. So it has to be the same for all of your test entries, for all of your experimental groups, so that only the independent variable would affect the response, nothing else. Okay. Now, so these is one important part. Okay, which is why I label it the key factors. First is your control. Okay, so this is another group or experimental unit that you assign within your experiment, but it will not be exposed to your treatment. So meaning it is under normal condition. Okay, no treatment here. Meaning, for example, in our study in rice, it is under just distilled water, no stress. Okay, why do we do that? Why do we need to have a control? So it serves as baseline for comparison with the treatment group. Is your treatment more effective than your control? Is your treatment less effective than the control? And you can also have a control experimental group or a control experimental unit. For example, if you're testing certain rice varieties for drought, so you can have a control of a known drought tolerant one and a known drought susceptible one. So you can see which one is better. Okay, so this is really important. You guys should have a control. Okay, regardless of whatever study you're doing, you should have this. If you don't have a control, then it will be hard for you to really uh, interpret your result. It will be really hard for you to say, okay, our treatment is better. Next is your replication. It is the repeated application of treatment under investigation. So usually you will read studies having the experiment was laid out with 3 replicates, something like that. So it is really important for you to assure the precision of your responses. For example, if you only have, this is a practical tip, if you only have one experimental group and suddenly something happened that is beyond your control, meaning wala ka na kaagad result, wala ka na kaagad experimental unit. If you have replication, you have another other two to get your data. Plus, you are making sure that, okay, are the treatments being imposed right? Okay, sorry, what was it? Okay, So again, you need replication to actually ensure that the results that you're getting are precise. Okay, that the responses of your experimental groups are precise. Okay, again, you need to have replication when you conduct a research, especially an experimental one. Next is randomization. So it is the spread of difference between subjects in random ways so that there will be no edge to one group. So this one will make you less biased. Okay, so ibig sabihin, when you uh, lay down, okay, your experimental group for either in the lab or in the field, you have to place them randomly or in a certain way, which we will discuss later on. So this is done to avoid being biased sa isang group. For example, you have your treatment, okay? You have, for example, treatment 1. Tapos nilagay mo lang siya, for example, sa isang side ng iyong experimental area or experimental field. If the condition there is conducive, yung test entries mo sa treatment 1, for example, will grow really good as compared to your other treatments and to your other experimental group. But if you randomize it, there is a chance for your other treatments to be on that region and some of your treatment 1 or other treatments to be in a less favorable environment. So you will have a grasp of the responses in the entire condition. Okay, so I hope medyo malinaw yan. And then, of course, like I said, it eliminates uh, sys- systematic influences upon assignment or selection. Okay, again, you need randomization. You don't have to be biased. You have to randomize it. Okay, so remember, you need to have a control. You have to replicate it for you to be precise, and you have to randomize it to minimize bias. Okay, so this is the three important factors I need you guys to remember. Next, okay, so this is what I'm talking about. So now we're talking about randomization and replication here. So let's talk about first the good designs. First is you have your completely randomized. So as you can see here, for example, he has two treatments here with 4 replicates. So as you can see, the experimental groups are evenly spaced, okay, and there's a random distribution of your replicates. So for example, if you have a gradient of stress, okay, so let's say the condition is not favorable, okay? So if the stress or the source of variation is coming from here to here, from left to right, meaning all of the experimental units and all of the treatments received the same amount of stress, and they were all exposed to the same amount of stress or sources of variation. Now you have your randomized block. As you can see here, it's in blocks, okay? But each treatment and is represented in each block. So this is still a good uh, design. So I will explain the difference of these two later, and we will talk about this deeper in the next slides. And then you have your systematic. So there is a pattern of how he laid down his experimental units and treatments. Okay, so these are the designs that you need to consider and that we say that these are good designs. Okay, now let's go to the poor designs. You have your simple segregation. So he segregated it based on the treatment. So all of his replicates 1, 2, 3, and 4 are on the left, and all the other ones are on the right. So this is not a good thing because again, if you are doing your experiment in a non-homogeneous environment, okay, meaning the conditions might be different, this will be biased. This is what I'm talking about. If the condition here on the left is better than the condition on the right, definitely the response on the left will be better. Okay. Next one is your clump segregation. Okay. So it's again separated and then there is a bigger space in between. So the responses here will be different. So if you do this, for example, in a field, let's say in the rice field, this group and this group will of course have better response. I mean, this specific replicates this one, the fourth one, and uh, the first one on the right. And of course, again, if you have a different sources of variation here, this will have different effect on that, and this will have effect on that. And this is one thing, the isolative segregation, this is again uh, one design you should uh, not do. Why? Because you're limiting the condition here inside chamber one, and you have another condition here inside chamber two. And of course, magkaiba pa ng treatment. So definitely there is an influence here playing. You have two influences playing here already. Then you have your randomized but with interdependent replicates. So nag-randomize ka nga, pero meron pa ring relationship yung bawat replicates mo. So para ka rin hindi nag-randomize dito. And then of course, the worst thing that you can do is to have no replication. Okay, so again, you need to have replicates. So now let's talk about the different experimental designs. So first, let's talk about the designs based on your experimental objective. Okay, so first you have your comparative objective. So make a conclusion about one factor from several factors, whether or not it is significant. Okay, so you wanted to conclude which factor is actually affecting much in your experimental group or in your study. So in this way, you need to compare different factors. Okay, and then come up with which one is actually significant. So again, you're comparing things here. Then you have your screening objective to select or screen out few important main effects from less important ones. So think of it as all of them actually have an effect. Okay, but which one has the main effect? So the first one, you have individual factors. Okay, the second one has an interaction here. Okay, so that's I think the one you should remember. And then the last one is optimizing responses objective. So you are selecting the best proportion of factors to maximize the response. So think of it as you're like cooking something. So anong recipe yung kailangan mong gamitin? Anong level ng factor 1, anong level ng factor 2, anong level ng factor 3 will give you the best result. Okay, so that's your optimizing your response objectives. Okay, now let's go to the basic and common types of your experimental design. So you have your completely randomized design, you have your randomized complete block design, split plot, Latin square, repeated measures, and factorial design. So let's talk about the 6 uh, experimental designs one by one. So I chose the 6 because these are the main experimental designs that we have, but there are actually subtypes of this. But of course, this is the first thing that we need to understand before we go to those subtypes. First, you have your completely randomized design. So the treatments are assigned to the experimental unit at random. Okay, so again, like the picture that I showed you a few slides ago, this is what it is. Okay, so ibig sabihin, random or walang order na sinusunod yung pag-impose mo ng treatment sa experimental units mo. So if you would lay this down, for example, you have your treatment 1 replicate 1, then you have your treatment 3 replicate 3, followed by your treatment 2 replicate 4, something like that. It is so random, okay? And then it allows all experimental units to have an equal opportunity of receiving treatment. So ibig sabihin, lahat ng experimental unit mo makaka-receive ng same ng treatment. Okay, not the same, of course, because you have different treatments, but all of them have an equal opportunity of receiving a treatment. Okay, so what are the advantages? It is completely flexible in the number of treatments and replicates. So kahit gaano karaming treatment, kahit gaano replicates, pwede. Kung gusto mo ng 10 treatment, fine. With 10 replicates, fine. Okay? So it has an easy statistical analysis even with missing data. So kahit na meron kang konting missing data, madali mo siyang ma-analyze, especially if you're gonna use software. And by the way, this is one thing that I wanted to emphasize. Your experimental design is really important especially what type of experimental design will you be using, because you have to input that when you analyze your data. Especially if you're using software, tatanungin niya kung anong klaseng experimental design yung ginamit mo. Saang experimental design galing yung data na in-input mo sa software. What are the disadvantages? Of course, low accuracy due to lack of restrictions, and allows environmental uh, variations to enter your experimental error. So, hindi mo pwedeng gamitin ang CRD, or there is a restriction in using CRD if there are environmental factors at play. For example, if you will do your experiment outside, that the conditions are not homogeneous, CRD is not the best option for you. Why? Because one part, okay, will be uh, affected, and you want. I mean, it might be okay if you think, but no, because the environmental factor will be at play here, and there will be an effect in your result, and you can actually see that when you analyze your data, because the experimental error will be high. Okay, so let's talk about what it looks like. So for example, here you have four treatments. Okay, you have treatment 1, 2, 3, and 4, and you can see here how randomized I actually um, placed all the treatments and all the replicates. Okay, so it's really randomized. There's no order here. Okay, so meaning all of your treatments are present in all of your replicates or experimental units. Okay, so this is your completely randomized design. So this is how you set it up. Next is your randomized complete block design. Okay, so take note of the word block. So experimental units are grouped into blocks, sorry, according to known or suspected variations, which is isolated by blocks. Okay, so within each block, conditions are homogeneous as possible. Okay, so for example, you have a source of variation. For example, if you're doing this in the field, I'm sorry, I'm always using field experiments because that's my thing. So for example, you have an irrigation system in one field and it's not leveled out properly. Mas malalim sa isang side, mas mababaw sa isang side. So you would actually have a blocking so that you have one block dun sa malalim na side and you have one block dun sa mababaw na side. Okay? And then all of your experimental units are there in one side, and all of your experimental units and treatments are in the other side as well. Okay? So what are the advantages? Again, it's also completely flexible in number of treatments and blocks. You can do as much as you want. It also has easy statistical analysis, even with missing data, and it allows you the calculation of unbiased error for specific treatments. Okay, so uh, kaya niyang i-rule out yung error, okay, without being biased. So the advantages is actually this is one disadvantage. So this is contradicting a little bit. So not for a large number of treatments. You can be flexible with the number of treatments, but you cannot go so large. Okay? Why? Kasi restricted ka ng blocks. Remember, you have all of your treatments should be within your blocks. So if marami kang treatments na nilagay dun sa block, magiging less yung replication mo. Magiging less yung randomization mo. Okay? So you have to keep that in mind. Then the interaction between blocks and treatment effects may increase error. So there is a chance that the blocks and the treatment can interact, or even the different blocks can interact. Okay? But this is a really good design, especially if you're doing your research in the field or outside. So this is how it looks like. For example, you have the variation gradient here coming from top to bottom. Okay, so you have block 1, 2, and 3. So just if you can see here, all of your treatments and all are in one block, and it has 4 replicates. The same thing in block number 2 and the same thing in block number 3. As you can see, there's also a different randomization in block 1 as compared to your block 2 and block 3. So this is how you should uh, set up your randomized complete block design. Okay, so there's actually an in between your randomized complete block design and your completely randomized design. So what people do is that this will be your replicate 1, this will be your replicate 2, and this will be your replicate 3. Okay, so that's one thing. That is if you have less experimental units to begin with. Okay, next is your split plot design. So this is for a multi-factor experiment using multi-experimental units. Okay, so you have different factors playing and you have different experimental units. So it is needed when levels of some treatment factors are difficult to change during an experiment. So ibig sabihin, hindi mo masyadong ma-contain or hindi mo masyadong ma-control yung factor na yon. And of course, if you cannot control it properly, it will have a huge effect in your experiment, which is why this design is perfect for that. So the levels of the first factor are applied to the experimental units with randomization. So first, you have to apply the factor in a random order. Okay? Hindi pwede na magkakasunod, naka-clump together yung treatments mo sa factor 1. Okay? Then the levels of the second factor is applied within your first factor. Okay? So then your replications and randomization of the second factor is within your first factor. Okay, so for you to visualize it, it looks like this. So for example, you have a treatment or a factor. For example, this is your N. Let's say it's your nitrogen level. Okay, so you have 4 factors here. Okay, and you have 8 plots here, 8 plots available. And then in each of your nitrogen level, you have G1, G2, and G3. Let's say these are your different varieties, or you can actually say that this is another, let's say, hormone application in plants. So as you can see, in treatment 3, nitrogen level 3, you have all of your other second factors. In your treatment number 1, you all again have it. Kasi mas madali mong makontrol itong second factor compared sa first factor, kaya siya yung ipapasok mo dun sa first factor. Okay? And of course, this will be on different plots. This is not block, it's plots. Okay? So plot meaning the area, the land area where you're using it. So your blocks can actually within a plot. Okay? So again, okay, so medyo nalayo yata ako ng konti. So just going back, your second factor is within your first factor. Okay? Next, you have your Latin square. So again, this is for experiments with 2 blocking factors. So you have 2 different sources of variation here, which is in one experimental field. So your experiments or your situations where there are 2 extraneous sources of variation. Okay, so you will have a row and a column here, which will have to be in a square plot. Okay, and wherein your treatments and experimental unit should appear exactly once in each row and column. So how does it look? I think it will be better if we have a visual representation of this. So this is how it looks. For example, you have a source of variation going from top to bottom, and at the exact same field, there's another variation going from left to right. So for example, this is your water level going from high to low, and this is, let's say, the gradient of your sunlight coming from east to west, or yeah, okay. So as I said, each of your treatment should be present in each row and each column. Okay, so as you see here, there's no walang treatment and replicate na magkatabi. Okay, sa isang column or sa isang row. Okay, so this is how you design a Latin square. Again, if you have 2 sources of variation. Okay, and again, it needs to be exactly a square. Okay, so it needs to be 4, it needs to be 16, something like that. A perfect square. Then your repeated measures. So each group member Okay, in an experiment, it's tested for multiple conditions over time or different conditions, meaning you only have one experimental group, which is you will apply the treatment and then paulit-ulit mong i-measure yung data or yung result. Okay? So you have different type of repeated measures. You have your ordinary repeated measures, wherein your experimental units assigned to single treatment and measured over time. So isang treatment lang yung nilagay mo. So for this is what I'm doing with my germination studies. So I will apply, for example, salt stress in my rice seeds, and then I will measure the germination of them every day for 7 days. So what will be the germination at day 1, at day 2, until day 7? Okay, so that is your repeated measures. You are repeating to measure, you are repeating the measure of one experimental unit over time, and you are only having a single treatment. Okay, the crossover design is that your experimental units are assigned in multiple or all treatments and are measured over time. Okay, so this is more applicable if you are conducting, for example, a clinical trial. So I'm not going to talk about the clinical trial because I'm not really familiar with it. But the point is, you have an experimental unit, okay, that you will measure, for example, 2 or 3 days under a treatment, a certain treatment, for example, treatment 1. And then you will transfer that experimental unit to treatment 2. And then now you're going to measure again. Okay? So the advantage of your repeated measure is that you need to have only fewer samples. Okay? So makakatipid ka dito. So how does it look? For example, you have 5 entries or 5 samples receiving the same treatment. You have time point 1, you have time point 2, going to time point 3, and time point 4. The time points, ikaw ang magde-decide nun. Are you going to measure every day? Are you going to measure every other day? Are you going to measure every week? So this is where your decision would come in. Okay. Next is your factorial design. So the influence of all experimental factors and their interactions or their responses are investigated. So you have multiple factors here, and you wanted to see what is the effect of their interactions in your experimental group. So it examines the main effect of 2 or more individual independent variables simultaneously. Okay, so lahat ng factors na in-play mo or na nilagay mo doon sa yung experiment, lahat yun titingnan mo at the same time. Okay, so how does it look? For example, you have a factor A here and you have a factor B. Your factor A has 3 levels or 3 types, whatever that is. And your factor B has 2 types or 2 levels. Now, your factorial design would actually let you see what is the effect of the interaction between your factor A type A and factor B type 1, and so on. Okay? So for example, here I have different nitrogen level. I have level 1, 2, and 3. And I have here 2 different stresses. For example, I have drought and I have flooding. So what is the effect of nitrogen level 1 in drought stress? What is the effect of nitrogen level 1 in flooding condition? What is the effect of nitrogen level 2 in drought stress and your nitrogen level 2 in flooding condition, for example? So you're going to see interactions of factors here, okay? And see which one is more effective, which one is more drastic, or something like that. Okay, now, what do you need to consider when you design or set up your experiment? So now you've known the type of your design based on the objectives and the type of design based on laying out your treatments and your experimental units. Now, what do you need to consider, especially when you're trying to decide, okay? How many replicates do you need? Do you need to have a control treatment, or do you need to have a control experimental unit, or do you need to have both? Do you need to have a negative control, a positive control, or maybe you need both? Okay, how many levels and increments of the stress of the treatment should you use? How many blocks do you need? And what type of design should you use? Okay, so these are the questions that you need to ask yourself before you start setting up your experiment. Okay, now these are what you consider. If you can answer these questions, you will have a clear view on what your experimental design should be. First, you need to understand the objective of your study. Why do you want to do the experiment? Ano ba yung objective mo? Meron ka bang factor na gustong pagkumparahin? Gusto mo bang makita yung effect ng certain level ng treatment mo sa experimental unit mo? Okay? What output or responses are you expecting? Ano yung ina-expect mo na ma-measure dito? Are you expecting an increase in height, for example, if you're dealing with plants? Are you expecting a huge zone of inhibition if you're dealing with antibacterial study? What is your hypothesis? Okay, so the first and most important thing is that you need to understand your objectives and you need to know your hypothesis. Second is you need to understand your study, the entire study, before uh, going into that. So did you fully understand your objectives? You know your objectives, but do you really understand it? Do you understand the background of your study? Okay, the reason why you're doing it, and do you understand actually the significance of your study? Will this stud- will Is it really significant, or what is the significance of it? So that is also important. Next, relevant studies. Have you read enough studies related to your research? Na-exhaust mo na ba lahat ng research papers, lahat ng books, lahat ng chapters related sa study mo? Okay? And have you found a research design or setup that is applicable to your research based on published reports? You can use this as reference actually. So, okay, this paper, their study is similar to what I want to do. Ano kaya yung ginamit nilang research design? Now, their research design may or may not be applicable to you. So if you have read a lot of uh, research articles, you know what to change, you know how to change their experimental design to fit your purpose or your research to get into your objectives. Okay, next. This is one important thing, the conditions of your experimental area. You have to know saan mo i-set up yung experiment mo. Uh, Again, saan mo siya i-set up? And is the area homogeneous? When we talk of homogeneous, it means the area has the exact same condition all throughout. All of the area has the exact temperature. It has the exact relative humidity. It has the exact sunlight coming in. And will there be external factors that can affect the response? Meron bang environmental factors or external factors beyond your control that could affect your result, that could affect your experiment? So just an example here, based on my experience, I set up a drought experiment in our greenhouse. Okay, but I failed to consider. Okay, I failed to consider uh, the current environmental condition. Okay, so hindi ko ina-expect na masyado palang mataas yung relative humidity sa loob ng greenhouse. Hindi ko ina-expect na masyado palang malakas yung pasok ng sunlight. So what happened? My drought stress became more stressed that the response of my positive control became negative. So, so that's the importance of knowing the condition before you actually establish your experiment. Okay? Next is the availability of materials. For example, you wanted to test your treatments on a certain type of plant, a certain expensive type of plant. So, ibig sabihin, konti lang ang magiging source mo. So, are your selected subjects available? Baka meron kang iniisip na experimental unit or subject na hindi naman pala available sa Pilipinas. Something like that. Or are your subjects right for your objectives? You wanted to use this type of plant, you wanted to use this type of bacteria, You wanted to use this type of animal. But yun ba yung tamang subject para makuha yung objective mo? For example, if you're going for an antifungal activity, are you going to use a bacteria? Definitely not. And then if you're going to use a fungi, what type of fungi? If you're going to use a bacteria, what type of bacteria? Is it gram positive? It is gram negative, or both? Okay. Next are the variables to consider. Okay. What important variables you need to get? Ano ba yung ina-expect mo na i-measure mo? Okay. Do you need to measure all of the things that's going to happen there? For example, if you're dealing with plants, do you need to measure the height? Do you need to measure the width? Do you need to measure the leaves? How many leaves are there? The sizes of the leaves? Do you actually need all of those? So that's one thing you need to consider. Okay. And then how are you going to measure these changes? Okay. Do you need to measure it using an equipment? Do you need to measure it using the common, for example, ruler or something like that? Are you just going to count? So those are the things that you need to consider as well. Okay. And of course, your resources. Will you be able to procure all of your experimental units? Baka naman masyadong mahal yung gusto mong subject. Baka naman merong alternative yon. Okay? Again, for example, if you wanted to use a certain expensive type of plant, maybe you wanted to go to something more common, something na hindi naman masyadong masasaktan yung bulsa mo. Okay? And will you be able to buy all necessary reagents if you're going to use chemicals? So if that in that case, you need to again search for alternative. Okay? Or you might need to cut down your treatments. Next is ethical concerns. Will you be using animal subjects? If you will be using animal subjects, you need to have the proper authorization and permit for that. So if you're going to use mice or hamster or rabbit, I think you need to get an authorization for that or a permit for that. Okay, and of course, health and safety. Are you planning on using hazardous chemicals like mutagens and carcinogens? Or is and if you're going to use those, is there a way that you can find a less hazardous ones that could actually give you the same result? Okay, so you need to consider that it's for your safety when doing the experiment. Okay, and so just a take-home message. Uh, when you consider, the most important thing is that you have to know the objective and you have to know why you're doing this. Okay, because again, your experimental design should answer your objective. Okay, so I guess that ends my presentation for today. So I hope you've learned uh, something about designing your research, uh, designing your experiment. Okay, so if you have questions, I'll be happy to answer them as much as I can. Okay, so do you have questions? Anyone? Okay, so
I think it was very detailed.
I'm so sorry. Was
Not
it?
even
Yeah, I think I went too overboard with that. Okay,
I think
so
it was very clear. Was it clear for all of you guys too? Like the visuals were very clear with all of the different setups. So
yeah, just one tip I think that I could say: if you're going to establish an experiment in the lab, I think the best option for you will be CRD. Okay, since the conditions in the lab naman are homogeneous enough, so that will be better for you guys. Uh, and also, I just thought of it like right now for the people who will be doing molecular studies. So especially for example sequencing and all that stuff. Uh, of course, the experimental design might not apply to them because that's a different type of research. But I think your replication can still apply there, especially if you're dealing with uh, mutated samples. So I think you still need to have replicate for that, at least three. If you're doing, for example, a gene expression study, you need to have three technical replicates with three biological replicates. Kasi you're dealing with numerical values if you're going to do some gene expression studies. So I think, yeah, you need to consider that. Okay. Do you have more? Do you have any more queries or questions? So, I mean, by the way, this lecture will be is being recorded. So if you have if some things are not clear to you, you can go back to the lecture and actually the slides will be available soon. So we will give you those so that you can review it if some things are a little bit hazy for you at this moment. Okay. Okay. So I think that covers the experimental design and setup. So I hope you guys have learned something, and I hope this could help you with establishing your experiments because December is now coming in, and I know you guys will be starting your researches now. Okay.